Great. Hi, everyone. My name is Elaine Shao. I'm an associate professor here at UCLA in the Department of Integrative Biology and Physiology. And today I want to talk a bit about the microbiome, which is of increasing interest to health and disease. So as overall background, the microbiome is really situated in large part in the GI tract. So the GI tract is the largest interface with the outside environment, spanning approximately 9 meters long, and has a really large mucosal surface area of about 32 square meters. And what's really interesting is just in the past 10 years or so, uh, largely from the NIH HMP project, we found that the GI tract contains many 100 trillion different microbes, about 100 trillion bacteria alone, not counting viruses, fungi, other microorganisms. And what's really amazing is that these bacteria contribute about 360 times the genomic content of even our own human genomes. It's also reported from the HMP that there are over 1,000 different bacterial species that have been identified. And just as a comparison, in the history of microbiology, there's only been about 30 or 40 so bacterial pathogens that have been studied. So this means that there's lots of new bacteria that are of great interest and of as yet unknown functions. At least some of the bacteria and other components of the microbiome are believed to perform really unique biochemical transformations, which makes them really important for understanding host health and disease. So aside from this and just the sheer number of microbes in the GI tract, there are also about 75% of the body's immune cells that reside in the lamina propria. And these immune cells are always actively surveilling the environment and interacting directly with the microbiome. And finally, there are lots of neurons in the GI tract as well, about 5 times more neurons than the spinal cord. And a subset of these neurons are believed to be chemosensory and to extend their axons directly up into the intestinal villi right behind the epithelium. And so overall, there being lots of microbes, a direct connection with the outside environment, lots of immune cells and lots of neurons suggest to the field that the microbiome is situated at the interface of gene-environment interactions and also potentially peripheral neuroimmune interactions. So what does this mean for epilepsy? I think for the microbiome in epilepsy it's kind of a burgeoning area of study. What is already known is that the microbiome interacts intimately with the immune system. And in doing so, it also mediates many effects of different environmental stressors on the host. And so these areas here where the microbiome and its interactions with immune infections or different stressors, and correspondingly, the possible associations between stressors and infections and epilepsy suggest that there may be potential role for the microbiome. This is an area, these arrows connecting to epilepsy directly haven't been well studied up to this point. Really only the immune connections and the stress connections with the microbiome itself. So I think this warrants future interest in studying whether changes in the microbiome in response to infection and in response to stress may contribute to the effects of these factors on epilepsy. What has been studied in a few reports so far is also in contrast that the microbiome may mediate effects of anti-epileptic drugs or interventions like diet or probiotics on epilepsy. And so what's been most well studied so far, but still really in early stages, is the potential interactions between the ketogenic diet, which has been used for almost 100 years to treat refractory epilepsy, and interactions between this diet and the microbiome. And so I'll tell you a bit about the data that's been published in this area so far. But the model here overall is that potentially the microbiome may, a dysregulated microbiome may mediate effects of infection or stress, or increase risk for infection or stress and downstream risk for epilepsy. And in contrast, different microbial communities may also mediate or contribute to positive effects of interventions on epilepsy. And so what my group has worked on in mouse models is the question of whether the microbiome can mediate effects of the ketogenic diet. So we used 2 mouse models, and in one of them, which was the 6 Hertz psychomotor seizure model, the other one was the KCNA KCNA1 knockout mouse model for SUDEP. And in both of these models, we found that the ketogenic diet, indeed, it shapes the microbiome, and that the microbiome is necessary and sufficient for seizure protection. Subsequently, we identified specific bacterial species and particular molecules that predicted seizure protection in the mice with over 94% accuracy. And finally, based on these bacterial species and molecules, we identified microbiome-based treatments that protected against seizures in mice. And so here with a microbiome transplant, with a live biotherapeutic or probiotic treatment, and also a microbiome-targeted pharmacological treatment. And so these were kind of the first, I think, mechanistic studies that suggest that the microbiome could contribute to seizure risk. More recently, a work from Peter Turnbaugh's lab at UCSF also identified a separate pathway whereby the ketogenic diet in producing ketone bodies like beta-hydroxybutyrate is able to suppress select members of the microbiome that are known to be pro-inflammatory. So in doing so, they report that the ketogenic diet through the microbiome is able to suppress inflammatory Th17 cells. And so these 2 mechanisms combined, one through a metabolite-mediated mechanism, and here through an immune-mediated mechanism, could contribute to effects of the ketogenic diet. One huge caveat is that both of these studies are conducted in mice, and so these are really just proof of concept studies at this point. And there are a handful of studies that have been done in humans looking at differences in the microbiome in patients with epilepsy and changes in the microbiome after the ketogenic diet. And what you can see here is that initially these studies report changes in the microbiome either at baseline or after the ketogenic diet. But as yet, there's very little overlap across these studies in the exact microbial signatures that have been identified. And so you may notice also these studies are quite small to this point. And so a big area of interest is also expanding this work to be more well-powered and more well-controlled in terms of controlling for factors that can influence the microbiome. And so I'll stop here and just say at this point, a lot is known about how the diet can shape the microbiome and how the microbiome mediates many effects of other diets on host susceptibility to disease. And I think it's really an intriguing area of future research to study how the microbiome and the ketogenic diet interact to influence epilepsy, or even to influence peripheral comorbidities that are still impactful for patients that suffer from epilepsy. And so I look forward to future discussion at the meeting. Thank you very much for your attention.